Antibiotic resistance is what happens when bacteria become insensitive to the existing drugs. This is a huge problem now. About 4.5 million people a year die of antibiotic-resistant organisms around the globe, and it's only getting worse. So our old drugs, mostly discovered in the 20th century, are becoming less and less effective as bacteria become more resistant to them. And so what we need to be able to do is continue to find new chemistry, new molecules that can actually address this problem, and do our best to bring them to patients in the future. It's hard to find new antibiotics because antibiotic discovery is hard. The bacteria themselves are marvels of evolution. They've been on the planet for 3.5 billion years. They know how to get around toxic molecules. So what we've been doing is looking for new molecules that we could bring into the antibiotic pipeline. So we ran a screen to identify new compounds that could address a big problem in antibiotic resistance. So we identified this new peptide antibiotic. It's a lasopeptide. What's interesting about this lasopeptide that we've discovered is that it binds to the protein synthesis machinery in a way that's completely different than other antibiotics. And it's not susceptible to existing resistance. That's a new molecule with a new mode of action. So this is a big leap forward for us.
The journey began with our extensive research to find antibiotic-producing bacteria. We collected soil sample from a backyard in Hamilton, and we let the bacteria grow on agar plate for about a year. One of these bacteria, called Penibacillus, was producing a substance that had strong activity against multidrug-resistant bacteria. Now, this was very exciting. This bacterium is producing something new, so it was a breakthrough moment. This is the 3D model of lyrosidin. It's like a small protein made up of 18 amino acids. As you can see in the green part, they are making a closed ring formation through a chemical bond, and the remaining tail of the peptide is threaded through this ring, giving this a unique 3D slip knot-like structure. Lasopeptides are extremely stable. You can literally boil them and they are still active. They are very resistant to proteolytic degradation by host enzymes. And due to these properties, lasopeptides are gaining increased interest in drug development these days.
So this discovery is absolutely astounding for us. This is a molecule that is not toxic to human cells, that is not susceptible to existing antibiotic resistance, and also works incredibly well in an animal model of an infection caused by a drug-resistant organism. Right now, this is a molecule produced by a bacterium, and bacteria are not interested in producing drugs for us. Drugs are really complicated compounds to prepare. So we're very excited about moving this project forward. So the initial discovery, which is the big aha moment for us, was astounding. But now the real hard work begins, and we're moving to take this molecule and working on ripping it apart and putting it back together again to make it a better drug candidate.